So hi, my name is Ana Takakura and I will be the host of this virtual journal club. Before we get started, I would like to thank the Journal of Physiology for this kind of invitation. And also I would like to thank the opportunity to discuss one paper. I also would like to remember that this session is being recorded and emailed to you in a week. And following this session, we will have a 30 minutes network session. And I would like to invite you all because I think it is a good opportunity for the attendees to meet and also to reflect about the paper. So the link for this session will be available in the chat at the end of this meeting. So today, the paper that we are going to discuss is the inhibitory control of active expiration by Botzinger complex in rats. I also would like to thank Dr. Daniel Zoccal for accepting to be a panelist, and also Dr. Ken O'Halloran for also accepting to be a panelist. So this virtual journal club is going to be divided in 4 parts. The first one that includes this introduction and also a little bit of the background related to the paper. And this is going to be my responsibility. So as I told you, I am Ana Takakura and I'm an associate professor in the University of São Paulo, Brazil. And I study the respiratory function in Parkinson's disease. In the second part, we are going to have the participation of Dr. Daniel Zoccal. He's also an associate professor at São Paulo State University, Brazil, and he studies the neural control of cardiorespiratory function in health, high altitudes, exercise, sleep disorders, and hypertension. And he is the corresponding author of this manuscript. In the third part, we are going to have the participation of Dr. Ken O'Halloran, that is a professor in the University College Cork in Ireland, and he studies the cardiorespiratory function in health and disease. So in this third part, Ken is going to give us an idea why he, as an associate editor of the Journal of Physiology, decided to accept this manuscript. And in the last part, we are going to have the questions and answers. So please, I would like you have the option to write your question at any time during this session using the Q&A button at the bottom of the screen. And in the end, we are going to have these questions that will be answered by Dr. Zoccal. So now let's go for the background. So the eupneic breathing is composed by 2 phases. The first one that is characterized by the contraction of diaphragm, and it leads to inspiration. And after that, we have a second phase that is characterized by the relaxation of diaphragm, and it calls post-inspiration or passive inspiration. Only in situations where we have an increase in the metabolic demand, like during exercise, low levels of oxygen, or high levels of CO2, we also can have a third phase that it calls active expiration, and it is characterized by the contraction of abdominal muscle. So each one of those phases are controlled by different groups of neurons located in the medulla. And today I would like to focus in the third phase and the region that is responsible for generating it. So the region calls parafacial respiratory group or parafacial lateral region. This is a conditional expiratory oscillator. And the first description of this region was performed by Jankiewski and Feldman in 2006. So in this paper, the authors showed that a transaction rostral to the facial nucleus did not change the breathing pattern. However, when they decided to perform the transaction caudal to the facial nucleus, it's clear that the active expiration was eliminated, but the inspiratory activity still remains. So in this paper, the authors conclude that the active expiration generator would be located around the facial nucleus. Today, we know that this region has at least two populations of neurons. One that is located a little bit more lateral, that calls parafacial lateral region, and as I already said, is involved in active expiration. And another one that is located a little bit more medial, that calls parafacial ventral region, or the retrotrapezoid nucleus, that is involved with CO2 sensing and also with the modulation of inspiration. So these two populations of neurons also have a phenotype, a different phenotype. So we know that the PFL is composed by excitatory neurons, and the PFV is also composed by excitatory neurons, but they are also positive for neuromedin and FOX2B. In 2011, Pagliardini and colleagues elegantly showed that this parafacial lateral region is consistently suppressed by inhibitory inputs. So in these experiments, the authors performed the bilateral disinhibition of PFL by the injection of biquiculin and strychnine. And it's clear that after this bilateral disinhibition, we have the activation of we have the generation of active expiration. In this paper, it was also possible to observe that this kind of response was only possible when the injections were performed right in the lateral region, where we can see the full circles, showing once more that in this region we have two populations of neurons. So after this paper, the question that comes was, what is the source of inhibition to PFRG neurons? We have some candidates that could be the responsible for this inhibitory input to PFRG. So these candidates involve a group of neurons that are involved in breathing, but also have inhibitory neurons that also have inhibitory in neurons. So some candidates that we have is the nucleus of the solitary tract, the medullary raphe, the pedunculopontine tegmentor nucleus, and also the Botzinger complex. So we did some experiments trying to investigate these inhibitory pathways from NTS and medullary raphe to PFRG. That's what I'm going to show you next. Very brief. So we performed experiments where we injected the retrograde tracer, fluorogold, in the PFRG, and we tried to observe if we would be able to find inhibitory neurons that directly project to PFRG. So the data showed that we had some neurons that were inhibitory and they directly project to PFRG. And we also tried to perform the same, to test the same hypothesis by physiological experiments, where we inject muscimol in the region of the NTS. So muscimol is a GABAergic agonist, trying to eliminate this inhibitory pathway and to observe if we would be able to see the active expiration generation. So the data showed to us that only at rest, we were able to see the generation of active expiration, but in a tonic way. So with this data, we could observe the presence of, we have physiological and anatomical evidences of inhibitory inputs from NTS to PFRG. So trying to investigate the inhibitory input from medullary raphe to PFRG, we tried the same approach. So we inject the retrograde tracer fluorogold in the PFRG, and we went to look at the medullary raphe to see if we would be able to find any GABAergic neurons that directly project to PFRG. So the data showed to us that only raphe obscurus and raphe magnus had inhibitory neurons that directly project to PFRG. When we tried to observe to test the same hypothesis by physiological experiments, so first we decided to inject asaporin in the region of the raphe obscurus, trying to eliminate the serotonergic input to PFRG and see if we would be able to generate the active expiration. So the data showed to us that no change was observed under rest conditions and under hypercapnic conditions when we eliminate the serotonergic neurons. So in the same group of animals, we also performed the inhibition of GABAergic neurons of this region by the injection of muscimol in this region. So we try to eliminate this inhibitory input to see if we would be able to generate the active expiration. And the data show it to us that after the muscimol injection in this region, we had an increase in the active expiration only during hypercapnia. And the same approach was tried for the Raphael Magnus. And once more, only with the injection of saporin, we were not able to find any change in the resting and hypercapnia-induced respiratory changes. However, when we inhibit this region with muscimol, once more, we were able to see an increase in the activity of abdominal muscle only during hypercapnia. So with this data, we know that we have inhibitory inputs from nucleus of the solitary tract to PFRG, directly to PFRG, and the inhibition of this pathway is able to produce a tonic activity in abdominal muscle. We also know that there is a subset of GABAergic neurons in the medullary raphe that directly project to PFRG, and this pathway is involved in the active expiration under higher levels of CO2. So knowing that, the next step, and that's what this manuscript that we are going to discuss today is the main question. So we want, Daniel and his colleagues decided to investigate if this inhibition to PFRG could come from the Botzinger complex. So thank you very much, Daniel, for your acceptance, and congratulations for your paper. The stage is yours.
So I'd like to start saying thank you very much, Ana, for selecting our manuscript to be discussed in this journal club. Thank you very much for that. It's a pleasure to be here and an honor to discuss this data. And also I'll say thank you to the Journal of Physiology and the Physiological Society for organizing this discussion. So in the following 15 to 20 minutes, I'd like to present the data related to this work, where we explore the role of the Botzinger neurons in the control of active expiration. Just one sec. Okay, so as Ana nicely demonstrated before, active expiration, which is a respiratory pattern, consists of contractions of abdominal muscles to force exhalation, is generated when we have the recruitment of a conditional expiratory oscillator that is located in the parafacial respiratory group, or more recently named as lateral parafacial region, to avoid some confusion in the literature. So this oscillator is located rostral to the ventral respiratory column and lateral to the caudal pole of the ventrolateral to the caudal pole of facial nucleus. So this oscillator is silent under resting conditions, under conditions of normoxia or in normocapnia. But when you have situations of elevated chemical drive, such as hypoxia and hypercapnia, this oscillator is recruited and generate active expiration. So there are some evidence in the literature indicating that this oscillator, some of these neurons in the PFRG are not sensitive to CO2, because some of them do not express FOX2B, and also some evidence that the blockage of glutamatergic transmission is able to eliminate active expiration during hypercapnia, but not during hypoxia. So indicating that, as Ana showed, that PFRG and the RTN neurons could be distinct populations of neurons, and indicating that synaptic inputs will be important for the recruitment of this oscillator. And then, as Ana also demonstrated, inhibition is important to suppress this oscillator under resting conditions. So we'll have at this point that probably excitatory and inhibitory inputs are important to control the activity of this expiratory oscillator. But we also have some evidence that inhibition is important for the pattern formation of the control of the PFRG activity and the pattern formation of abdominal expiratory activity. So this is an example of extracellular recordings of neurons in this parafacial region. Under baseline conditions, no activity is observed in these neurons because they are synaptically suppressed. But during elevated chemical drives, such as hypercapnia, these neurons start firing rhythmically during expiratory phase. So we still don't know if this activity relies on intrinsic mechanisms, but we have some suggestions based on the mathematical model that will have a balance between excitation and inhibition to control this, the activity of these neurons. So according to the model, we have in the presence of excitation driven by hypoxia or hypercapnia, inhibition during inspiratory and post-inspiratory phase are important for the pattern formation of these late expiratory neurons, where potentially pre-Botzinger neurons, inspiratory neurons provide inhibition during inspiration, and Botzinger neurons provide inhibition during post-inspiration. So these suggestions came from mathematical simulations. So in this paper, we decided to investigate the role of the Botzinger neurons in this control of active expiration. So to explore this, we perform experiments in the celebrated arterially perfused in situ rat preparations. And we chose this, this preparation because of its advantage to perform multiple recordings from respiratory motor outputs in the absence of anesthesia. And also this preparation, in this preparation, we'll have easy access to the respiratory network, which make our life easier to perform microinjections as well as position electrodes to record the activity of the respiratory neurons. So in these experiments, we recorded from vagus, phrenic, and abdominal activities, and we perform recordings of the Botzinger neurons, extracellular recordings. We also perform microinjections of agonists and antagonists in the Botzinger PFRG, as I'm going to show later in the results. In these preps, we induce active expiration using hypercapnia. So using preparations of naive animals, we expose them to hypercapnia to trigger active expiration. And we also use a model of animals previously submitted to sustained hypoxia, 10% of oxygen during 24 hours. And this is an interesting model because at this condition, animals exhibit active expiration under normal conditions without the need for elevated chemical drive. So at the bottom, we have some tracings illustrating the pattern of activities of abdominal and phrenic under baseline conditions and in the presence of active expiration, where we can see these bursts in abdominal, characterizing the pattern of active expiration. And this data, the experimental data that we generated, we incorporated in a mathematical model to gain some information about potential connectivity among the neurons, the Botzinger neurons and the PFRG as well. So we start this work recording from the Botzinger neurons to have an idea what's going on with the activity of these neurons when we have the presence of active expiration. So within the Botzinger neurons, we have two populations. We have post-I, post-inspiratory neurons, and AUG, augmenting expiratory neurons, which are characterized by their firing pattern that I'm going to show you in the next slide. So basically in this protocol, we identify the neuron using an electrode, record their activity under normal capnic and under hypercapnic conditions. So on the right here, I have a representative tracing illustrating how we perform the experiment. And then, so here we have phrenic, abdominal, and in this case, an augmenting expiratory neurons in the Botzinger. So we recorded the activity during normal capnic conditions and during hypercapnia in the presence of active expiration, noticed by the presence of this lady burst in abdominal. So here we have an expanded tracings of abdominal, phrenic, and post-I neuron in the Botzinger. So the post-I neurons are characterized by a decrementing pattern of discharge during the expiratory phase. So we can see action potentials until the very end of the expiratory phase under normal capnia conditions. But in the presence of hypercapnia, and when we saw lady bursts indicating the presence of active expiration, we noticed that the post-I neurons stopped firing before the emergence of active expiration. So suggesting kind of negative relation with when you have the presence of active expiration, post-I neurons are silent. Recording from augmenting expiratory neurons. So these neurons are characterized by an augmenting firing pattern of discharge during expiratory phase. Usually these neurons start firing at the mid of expiration, peaking at the end of expiratory phase. In the presence of active expiration under hypercapnia, we notice a slight early onset of algae activity within the expiratory phase and a higher discharge frequency during the coincident with the presence of lady bursts in abdominal. So suggesting that these neurons could be excited in the presence of active expiratory pattern. So these recordings showed us that Botzinger and algae neurons are distinctly modulated during the emergence of active expiration. Later, we decided to play pharmacologically with the Botzinger neurons to gain information about their functional role in controlling active expiration. So it is suggested that post-I and algae neurons interact motilely through inhibitory synapses using glycine and GABA as neurotransmitters. So we decided to manipulate the systems and to change the activity of these neurons and see what happened with active expiration inducing during hypercapnia. So our working hypothesis was that during hypercapnia, when we have an increased excitatory drive from central chemoreceptors, here illustrated by the RTN chemo drive, we have excitation of parafacial region, which then excited algae neurons, which in turn inhibited post-I and then send an inhibitory inputs to the PFRG, contributing to the pattern formation of these neurons. So in these experiments, we decided to perform disinhibition of the Botzinger by microinjection strychnine bilaterally in the Botzinger complex, because we considered that these could increase the activity of the post-I neurons and then enhance inhibition to the PFRG, which would be enough to suppress active expiration during hypercapnia. So here is the experimental data. In this upper panel, just to show that we use a low concentration of strychnine, which caused minor effects on the baseline breathing, but did not disrupt the free-phase respiratory pattern. And here you have the recordings under hypercapnia. So here you have before injections, presence of lady bursts, presence of active expiration. And this pattern was suppressed after strychnine microinjections in the Botzinger, which somehow agree with our hypothesis that glycinary transmission control neurons that suppress, that inhibit PFRG. It's important to inhibit PFRG under conditions of hypercapnia. Following the same rationale from previous study, we perform these experiments, this inhibition of the Botzinger now with microinjections of gabazine, because we were planning to block this GABAergic transmission, which could cause a similar effect, increase the activity of post-I neurons and then suppression of PFRG. But surprisingly, when we did these injections, we observed an opposite effect. So here in the tracings, we have these arrows indicated the moment of injections of gabazine in the Botzinger. And after the second injections, we notice the presence of active expiration under normal capnia. I mean, no chemical drive. So under baseline conditions, this inhibition of or the antagonism of GABA receptors was able to evoke active expiration. So we have expanded recordings demonstrating the active pattern before and after gabazine injections, and we can see the presence of active expiration after the antagonism of GABA receptors in the Botzinger. So indicating that our working hypothesis was wrong, and then the mechanisms controlling active expiration from the Botzinger would be more complex than we originally thought. So with this complexity, we decided to use mathematical model to propose some possibilities that could explain how Botzinger could interact with the PFRG and control active expiration. So after discussion about the possibilities, we came with this new organization of the Botzinger complex, where we suggested two populations of post-I neurons in the Botzinger. One GABAergic, and here represented in green, and another one glycinergic, represented in magenta. So these two populations would send projections to the PFRG, so they are both responsible for inhibiting PFRG. And both populations require excitatory drive to operate. And we suggested that based on previous study that this could come from the pons. So the idea in this model is that this GABAergic population send projections to the Golgi neurons, and these glycinergic post-I neurons receive glycinergic inhibition from the Golgi population. So before we have two populations mutually interacting, and now we have a specific pattern of interactions between two populations of post-I. So this was the new idea about this, new possibility about the organization of this Botzinger complex controlling PFRG activity and the generation of active expiration. So with this model, with this configuration, we were able to reproduce some of the data that I showed before. So here we have the model performance. So these lines represent the average member quotation of these different populations of neurons. Here you have early I population in the Botzinger. So we have the inspiratory phase. We have late E activity in the PFRG, both post-I population in the Botzinger, glycinergic and GABAergic population, and here the Golgi population in the Botzinger. So this is the model performance under normal capnic conditions. So late E is suppressed, so no active expiration. And we simulated hypercapnia in the model by increasing the excitatory drive from the RTN to the PFRG. And by doing that, we saw the emergence of parafacial activity, represented here by the late E bursts. By doing that, we have this excitation of Golgi population, demonstrated here. And then we have inhibition of the post-I activity, decreasing the inhibition to the PFRG, which is important for pattern formation of the PFRG activity. So using this configuration, we were able to reproduce what we saw experimentally. Then we also simulated the condition of inhibition or antagonism of glycinergic receptors in the Botzinger. So here we have the model performance during hypercapnia with the emergence of active expiration, notice here. So we simulated the strychnine experiments by reducing the synaptic weight from the Golgi to the post-I neurons. And by doing that, we disinhibit this population, increasing the inhibition to PFRG. And by doing that, the model showed that this was enough to suppress PFRG activity even under hypercapnia, which agreed with what we saw experimentally. And the model was also able to reproduce the gabazine experiment. So we simulated that condition by reducing the synaptic weight between GABAergic population, post-I population, or GABAergic inputs, to the algae. These release these neurons to fire more, increasing the inhibition to the post-high population, reducing the inhibitory drive to PFRG, releasing these neurons to fire. And this is the model performance indicating that, that the reduction of the GABAergic synaptic weight to the algae neurons were able to increase their activity, suppress post-high activity, and generate active expiration under resting conditions. So with this model, we decided to explore more about these connections. And to do that, we now use the model of sustained hypoxia to generate active expiration. So as I said, these consist of animals exposed to 10% of oxygen for 24 hours. And after that, when the animals returned to normoxic conditions, they show active expiration under resting normoxic normocap conditions. So it's an interesting model of active expiration in the absence of elevated chemical drive. So in the in situ preparations, we have this type of recordings as we saw lady bursts under normal capping conditions. And we recorded from post-eye and algae neurons in these animals, and we noticed and we saw the same pattern of activity as reported under hypercapnia. So for post-eye neurons, we noticed this inhibition or no activity in the presence of lady bursts. So these recordings in sustained hypoxia animal. So suggesting this inhibitory or negative relation between active expiration and post-eye activity. And for algae neurons, we also noticed this increased discharge frequency in the presence of lady bursts. So as I said, similar pattern to what we observed under conditions of hypercapnia. And in the model, we mimic this condition of sustained hypoxia by, as I said, the chemical drive is the same, it's not changed. So we mimic sustained hypoxia in the model by reducing the activity of post-eye activity, both populations. And we did that by reducing the drive, the excitatory drive to these populations, as demonstrated here. And as a result, the inhibitory drive to PFRG is reduced. Then these neurons are under less inhibition, so they are able to fire under resting conditions. So using this configuration, this sustained hypoxic configuration, we then try to generate some predictions, again, as I said, to test if the model is working properly or not. So the first simulation that we generated was that if the sustained hypoxia model consists of a reduction of post-eye activity, if we, for some reason, generate a transient excitation of the Botzinger region, we would be able to suppress active expiration. So this is the model performance of sustained hypoxic model before and during this transient excitation of the Botzinger. And we can see this suppression of PFRG activity. So this was the prediction, and then we test this experimentally by performing glutamate injections in the Botzinger of control and sustained hypoxic animals. So in a control animal, we noticed some respiratory changes induced by glutamate by this response in phrenic and vagal activities, but at the abdominal activity, we did not observe significant change. In sustained hypoxic animals, on the other hand, we noticed that glutamate injections suppress active expiration. So the same pattern of activity that we predict from the model. So suggesting that the experimental and the model results, they are walking together. Then another prediction that we generate in the model is that the increase of, because we simulated that sustained hypoxia is dependent on the decrease of post-eye activity. So we test if we increase only the activity of glycinergic population in the Botzinger, it would be enough to suppress active expiration. So To do that in the model, we reduced the inhibition from Augie to post-I, which was enough to increase activity, post-I activity, as represented here in the tracing. And this was enough to suppress active expiration. So only manipulating the post-I activity, the glycinergic post-I population. So we test this experimentally again by performing microinjections of strychnine in the Botzinger of sustained hypoxic animals. So these are the tracings before injections. So you can see active expiration. And after strychnine injections in the Botzinger, we suppressed active expiration. So again, this experimental verification agreed with model predictions. And finally, we explore a little bit about inhibition at the PFRG regions. So previous study demonstrated that PFRG is suppressed under resting conditions. And this study mostly used a combination of GABA and glycinergic and glycine antagonists in the PFRG, or only inhibition of a GABA input to the PFRG. So in this set of experiments, we decided to focus on the glycinergic input to PFRG. So in the model, when we blocked or reduced the glycinergic inputs to PFRG, only this manipulation is enough to trigger active expiration under resting conditions. So just reducing the activity of the post-I glycinergic population. We also checked this experimentally. So we perform in naive preparations injections of strychnine in the PFRG. So this is before and after the injections where the antagonism of glycinergic receptors was able to trigger active expiration in the resting conditions. So finally, just to verify the contribution of both GABAergic and glycinergic inputs from the Botzinger, we decided to use this condition of decrease in the glycinergic inputs to PFRG and generate a transient excitation of the Botzinger. The idea was, if both connections are present, suppression of just one arm wouldn't be enough to eliminate active expiration. So this is the model performance. So we have recordings of the neurons in the presence of reduced glycinergic inputs to PFRG. We have active expiration in the resting conditions, and the transient excitation of the Botzinger caused some attenuation of active expiration, but did not eliminate it. We have some bursts during this period. So because we have two different inhibitory pathways. So we also test this experimentally. So this is the condition before we have the strychnine injections in the PFRG, generation of active expiration, and then we inject glutamate in the Botzinger. And we saw a reduction in the active expiration, but not an elimination. So confirmed, or at least indicating that maybe you have two different pathways, one glycinergic and another GABAergic, potentially controlling PFRG activity. So with this, we conclude that we have this new organization, microcircuitry in the Botzinger controlling active expiration. So our data support that Botzinger neurons are important or are part of the inhibitory circuitry that controls active expiration. Of course, there are other regions controlling that, as Ana showed at the beginning. But in this study, we are supporting a role for the Botzinger neurons. We are aware that most of our suggestions in this model are hypothetical, and we still have to do some work to prove some of these connections. But it's important to mention that some recent works demonstrating the presence of GABAergic and glycinergic cells in the Botzinger that send projections to PFRG, which support part of our connections that we propose in the model. But as I said, we still have some work to do to prove some of these connections. And our work is also important to demonstrate that these mechanisms in the Botzinger, they can go under plasticity in conditions, for example, of chronic challenges such as the sustained hypoxia. So these mechanisms would be important to be investigated as to understand the breathing changes associated with exposure to chronic hypoxia. But these are future studies as well. So I'd like to finish by acknowledging the authors of this study. First, I'd like to acknowledge and thank Karini for his brilliant and hard work in this study. She did most of the experimental part of this study. And also thank William, postdoc in Slava lab in the Georgia State University, which both did the model simulations. So thank you guys. And thank you, Dr. Mariluza as well. She contributed to this work by doing some recordings in the neuronal recordings sustaining hypoxic animals. So thank the authors and thank you for the financial support from FAPESP, CNPq, and NIH that gave us the money to run these experiments. So thank you all for your attention, and I'll be happy to take any questions.
Okay, thank you very much, Daniel, and congratulations for your beautiful data. And now let's go to the third part. So please, can you tell us why you decided to accept this manuscript? And also, we would like to know what you think it's important for getting a manuscript accepted in the Journal of Physiology. Thank you for your participation.
Sure. Hello, everybody. I'm conscious of time, and I do want to leave time for the panel discussion and for some questions related to the paper. But briefly, I guess Daniel has illustrated really the strength of the work. The piece that was most attractive about this particular paper was the combination of approach, the experimental approach, and the mathematical modeling, and the flexibility that's afforded by that approach if you remain as an investigator open-minded. So it shouldn't have escaped our attention that Daniel effectively threw out their first guess at it and said, well, that's not what the data show. We've got to go back and look at the model. We've got to work the model to give us predictions that we ourselves and that the field can follow through on. And so it was very much and remains very much a bidirectional approach. That's the great strength. If you think about neural circuitry and the complexity of the neural circuitry in this space, and I'm only on the fringe of this field, and it seems every time we think we've worked it out, it gets more complicated. And that's, of course, wonderful for new investigators coming through and even seasoned investigators to continue to sort out this puzzle. But that was evident at the outset. So it goes to a senior editor, it goes to me. We have a look at whether we think it's disruptive, whether it's going to change the way the field looks at this problem. And then, of course, we take expert opinion, and reviewers were sought, and a potentially difficult task in such a complex paper. And on the strength of those opinions then, and Daniel won't mind me saying that a thorough review is one in which further challenges are offered up in the course of peer review, and the journal, of course, prides itself on that, on seeking to publish very high quality work. There might be a sense sometimes that it's a little bit of a lottery when we get our papers through, whether it gets a good review or a bad review and so on, but many papers that are published in the Journal of Physiology are ones in which there's been a very thorough critique at the level of peer review, at editorial level and at the level of expert opinion. In other words, multiple iterations. And I know Daniel is open-minded enough to know that this work remains open to further investigation, further challenge, and its true impact will be seen maybe sometime down the road. I think it's perfect, in fact, that any good paper like this offers us more questions than it does some solutions. It should be a point of pride, actually, that you say there are now many testable hypotheses that you're interested in and that the field are interested in. In fact, you've gifted those because they are now for others to test as well and to piece back into the puzzle. And the model remains open then, if you like, for experimental evidence that can come back to the model, and the model will continue to make good predictions. I don't think a problem of this complexity could be solved purely experimentally. Perhaps if everybody in the field turned their attention to the Botzinger complex, perhaps so, but The model offers, of course, wonderful efficiency as well. It's dependent upon the experimental data, but of course it also shapes the direction of the experimental data. So all of those things and the impressive presentation of it today, and we should look forward to its how well it will be received by the field. All of that shapes why this is a paper that merits publication, and publication in a journal that prides itself on publishing the best work in the field. So congratulations.
So thank you, Ken. So let's go to the question and answers. So we have a question from Thiago Moreira from the University of São Paulo, Brazil. So he says, congratulations on such nice data. Why do we need so many inhibitory pathways to PFRG from different areas? So what's your opinion about that?
That's a good question. And to be honest, we still don't know why we have so many inhibitory players in this circuitry. Of course, it may depend on the situation, because we still have to understand when active expiration is important, because all the time that we explore active expiration, we use high intensity stimulus, like high hypercapnia, high hypoxia, and we don't know exactly when active expiration is triggered. So sometimes, because active expiration is cost energy, depending on the situation, we don't need that. So maybe these inhibitory pathways would be important to suppress active expiration when it's not needed. But of course, it will depend on the situation. Maybe we may have peripheral information, like from the lungs inhibiting active expiration, or we have just a regular inhibitory mechanisms operating every cycle. So I don't know exactly how to answer this question, but I think it would be in these lines too, but we still have to work on this to have more information.
So just thinking about this question, do you think that, for example, it depends on the kind of situation that we have? For example, if we have a hypoxia, hypercapnia, we could have a different pathway that would be suppressed. What do you think?
Yeah, no, I think this is the point. For example, if I can give you, I know there are differences between experimental approach, but in the in situ preparation, for example, when you stimulate peripheral chemoreceptors, we see active expiration, but we see a reduction in the expiratory time. While during hypercapnia, we have active expiration and the prolongation of expiratory time. So you have two different types of active expiration. So maybe it could be, so these inhibitory pathways could be important to shape how active expiration will be generated to have a specific respiratory pattern response, like an increasing frequency, respiratory frequency, or an increasing tidal volume. So I think depending on the situation, we'll have different pathways being recruited.
But do you think that, for example, Botzinger complex would be involved more in hypoxia than hypercapnia?
Good question. I don't know. This is something we need to explore, but it's a good point.
And I don't know, probably NTS can also be more involved in hypoxia than hypercapnia. I don't know.
No, sure, you're right. It's a lot of possibilities to test. You know. Thank you.
Okay, so our time is already gone. So once more, I would like to thank the Journal of Physiology for the invitation, and also all the attendees that we had today. Thank you, Daniel and Ken, for participating as a panelist. And once more, I would like to invite you all to this next 30 minutes network session, where we can still discuss a little bit more about this paper. So the chat should be right now in the link should be right now in the chat. Yes, it's already in the chat. So thank you very much, and see you in the next session.